Just send it.
Is it okay if I don't stay here? I could stay over there, but they don't need to see me, right? Because
They want to be.
they want to see.
Yes.
Okay.
You can use the pointer over there.
Yeah, I understand.
I think that should be fine. Please feel free to.
Yeah, but it's just too short so to go down. That's the reason.
No, that's perfectly
Yeah.
fine.
The Wi-Fi doesn't work.
Yeah, that's what I know.
I could link it to my phone. The problem is that will be slow.
Yeah,
His photo. His photos. Once he starts his photos, he's very upset.
ನಾನು ಲೊಕೇಶನ್ ಆಯ್ತಾ ಅದು ಮಾತಾಡಿ ಕಳಿಸಿದ್ದಾರೆ ಯಾಕಂದ್ರೆ ಅವರು ಮೀಟಿಂಗ್ ಸ್ಟಾರ್ಟ್ ಆಗ್ತಾ ಇದೆ ಅದಕ್ಕೋಸ್ಕರ ಓಕೆ
So can you just share your password? ಆಯ್ತಾ?
ಒಂದು 5 ನಿಮಿಷ ಅಂದ್ರು ಮಾಡ್ತಾ ಇದ್ದಾರೆ ಅದಕ್ಕೆ ಸುಮ್ಮನೆ ನಿಮ್ಮ ಲ್ಯಾಪ್ಟಾಪ್ ವೈಫೈ ಕನೆಕ್ಟ್ ಆಗಿದೆಲ್ಲ
ಯಾರು?
ಯೂಸ್ ಮಾಡ್ಬೋದು
open. With us today, Professor Gopalakrishnan. Bachelor's in pharmacy, and then he did his the further work in PhD by research at IIT Madras at masters, and then he went on to do his PhD where he brought his interest largely in intracellular cytoskeletons using electron microscopy and many other components, and from there on his interest has been in building organoids, brain organoids from the iPSC derived. And for the most fascinating publications which have come out is where he has been able to bring together the brain organoids with ability to have even the eye generation on those. That has been amazing, and that has changed the field by itself. Something that he has done. His large interest using the organoids is to build models for many human disease conditions, could be in Alzheimer's, even glioblastoma, and many other aspects. And I think it would be fantastic on behalf of CCAT as well as Eyestem, where they are discussing some of the potential work together. It's a great pleasure to have Jay with us. Warm welcome, and looking forward to your hearing your work and engaging more. Thank you so much.
It's my great pleasure to be here, and thanks for the complete Eyestem group, Vijay, Rajeshri, Jogin, sorry, it's you, and yes, I mean, I didn't sleep for the last nearly 30 hours, so that's the reason I'm just caffeinating myself. So what I'm going to today present is, I would like to introduce you about our earlier work, and then eventually I'm going to speak much more about the human brain organoids and how human brain organoids can help dissecting the human specific mechanisms. Therefore, we can address some of the human specific diseases such as microcephaly and potentially glioblastoma and so on. So you're able to see my cursor. Hold on. Yeah, so the research concept of my laboratory is to study the centrosomes and cilium biology. And then eventually they translate those basic understanding into clinical applications. And in this aspect, particularly, we are interested in human brain development, and then study the microcephaly, which is a developmental disorder where the neurogenesis is perturbed. And then we are also attempting to translate our finding to glioblastoma multiforme. Well, we know that human brain is one of the most complex tissue systems, and partly because it has got numerous kind of cell types collectively together to form the human brain, neural epithelial tissues. So one of the most important aspects that the human brain differs from the non-human primates, basically our cousins, great apes and the old world monkeys, that extended cortical surface and increased neuronal density. So these two aspects are dramatically differing from the non-human primates. So now that basically fascinated us to study the human brain development. Now, if you look at, this is a schematic diagram. So if you look at the human neocortex development, and you can see at the apical side, you have neural progenitor cells. These are all called apical neural progenitor cells. They give rise to different kind of NPCs. There are different kinds of NPCs. By symmetric and asymmetric division. And finally, these cells gets differentiated into cortical plate where you see the neurons. And this is a complicated process, and it's a highly orchestrated process. And in our opinion, there are a couple of bottlenecks to understand this process. Number one, the fate determinants, the regulatory factors that can determine these cells to either proliferate or differentiate. And then even if you identify them, there is no proper model system to study them. So what we think is that the centrosomes and cilia are one of the fate determinants in determining the apical progenitor's fate to differentiate into different kinds of neurons or neural stem cells. And second, we think we can address these issues by using human relevant system, which is basically 3D human brain organoids. Now, let me introduce you about the centrosomes and primary cilia. So this goes back several years ago. So the centrosomes are fundamental cellular structures, which are important for accurate cell division. And you can see the centrosomes have got two other structures inside it. It's called as centrioles. And these centrioles are microtubule-based structures. This is a mitotic cell. So when the cells are not in mitosis, the cells, now this is the mature mother centriole docks to the plasma membrane and assembles the so-called primary cilium. The primary cilium is traditionally known to be a cellular antenna, receives the extracellular signaling, titrates them, and transduces the required amount of signals into the cell for different gene expressions. And pretty much every single vertebrate cell types have got primary cilium. And this is electron microscopy image. You can see the mother centriole that assembles primary cilia, which is a microtubule-based structure, and the daughter centriole is just nearby. So you can see the centrosomes and cilium are very important for organism development. For obvious reasons, you cannot mutate them. You cannot eliminate them in mammalian cells or a living animal because they're important for animal development. And that's where the Drosophila came into the picture. We took Drosophila genetics as a model system because flies can develop without functional centrosomes. So during my postdoc work, we focused on, we used reverse genetics combined with the biochemistry. We cloned numerous centrosomal genes. And back then, we did not know what these genes are actually doing. And the flies can completely develop, but you can see they don't move and they are uncoordinated because they lack the sensory cilium, which emanate from the primary, from the centrioles. So by using the Drosophila as a model system, we contributed key works in the field on understanding the centrosome biology. All right, now let me move on to model systems, which I promised you. First, so far I was discussing about centrosomes and centrioles, some historical background. Now I'm going to move to the model systems. What is 3D human brain organoids? Why do we need it now? So brain organoids are self-assembling 3D structures. Basically, we exploit the self-assembling properties of the stem cells. And then by instructing them to allow under controlled differentiation into neurospheres, eventually to human brain organoids. These human brain organoids about 3 to 4 millimeter in size, and morphologically, the surface level, they mimic several aspects of the developing human brain. This procedure has been established in my laboratory and has been developed by other laboratories. Now it's widely used in several laboratories across the world. Now, if you generate brain organoids according to our method, which does not include EBs, for example, therefore you get a pure neural cell types. And if you section and if you analyze them, and you can see the ventricular zone, that's where the neurogenesis begins. And I'm just matching this to the schematic diagram. That's very important to compare it. And you can see at the apical side, you have a neural progenitors. On the basal side, you have a primitive cortical plate where you see neurons. What is important is that you don't want to see the mixture of these cytoarchitectures, apical to basal. That is already a disease phenotype. So this method allows to generate brain organoids in a how you expect to see from the developing human embryo. Thanks to the brain organoids, and since last 3 years, we have seen two epidemics and two epidemics, basically one pandemic and epidemic. And brain organoids have helped tremendously to address the infection mechanisms of Zika virus, as well as lately we showed that SARS-CoV-2 can enter into the human neurons. And basically our lab was the first lab to demonstrate, and we were very careful to make this point, that the virus can enter into cortical neurons in the human brain organoids. And this has been, again, verified by several clinical work. Okay, so so far I was talking about the cellular structures that we are interested in, and some introduction I gave you about brain organoids. Now the field is going very fast, and to keep ourselves competent in this lab, in this field, so we also wanted to generate brain organoids in an advanced manner. So that's why I wanted to provide you some examples for the next generation brain organoids. So we asked a series of questions. Now, how far can we make it complex? Remember, brain organoids are reductionist approach. So can we make it much more complex? And then we ask a question that how far the brain organoids genuinely recapitulate the human biology. And can we also generate so-called brain organoid factory, which I would like to explain that to you in a minute. Now, if you then look at the embryology book, and it's very clear. I mean, these works are like nearly 100 years old work. Like, the human brain develops from the neural tube, which is basically a monolayer neural epithelial cells. These cells differentiate, give rise to forebrain, midbrain, hindbrain, and spinal cord. All right, from the forebrain, other processes are developed, basically diencephalon. From diencephalon, there is invagination happens that gives rise to optic vesicle. Eventually, that would be optic cups, and then other cell types derived from both surface ectoderm, neuroectoderm, and the non-neuronal cell types, basically. They are assembled to form the human eye. So if the human brain organoids are really useful tissues, and if it recapitulates the human biology, we should be able to instruct these human brain organoids to generate optic vesicles, but in a precisely in numbers, and also topographically restricted in a manner that you don't want to generate more than 2, but only 2, but it should be also located in one of the poles. Otherwise, these brain organoids, chaotic systems, they don't even know how to pattern their, the patterning programs during the forebrain development. And we also expected that if this is really true, the brain organoids would also generate optic vesicles containing retinal pigmented cells, RPEs, retinal pigment epithelia, as well as lens structures and cornea structures. So these are all not from the surface, these are not from the neuroectoderm, but from the surface ectoderm. So we embarked on this question and we worked on this. So at some point around day 60, we identified these brain organoids developing these 2 pigmented dots. And that was already several years ago. That was very exciting for us. And then we wanted to analyze them further in detail. I'm not going to go much into detail, but what I can point out, if you do the flat embedding of these tissues and if you label them, and you can see these honeycomb-sized shaped structures, which are RPEs, retinal pigment epithelia. And every single cell has got a primary cilia and the apical site. It also has these structures have also got, these objects have also got lens-like structures, which are positive for alpha crystalline, and we have performed electron microscopy images to show that. And we wanted to also test if they have a photosensitivity, and they do have. And what you see is that hyperpolarization and depolarization, depending on the light intensity that you shed it. But one has to be careful here. These organoids are still in the early developmental stage. The light response that you see, not necessarily from the photoreceptors, because the photoreceptors are not even developing fully mature here. So we think it is because of the GOT2 positive, intrinsically active retinal ganglionic cells. But still, we need to analyze them in further details. And of course, if you do have questions, you can ask me. Yes.
So these 2 eye-like structures always formed at the same position?
Yes.
And if so, what was the molecular change?
Well, I would phrase it differently. Why should you expect to see these 2 optic vesicles in one side? Because that's where the forebrain entity is present. We then, in the manuscript, we described it. So if you do analysis, and you can see the FOXG1 gradient, basically, and this is one of the forebrain patterning molecules. Not only FOXG1, but several other forebrain patterning molecules are segregating and to define the eye field. And I think that's what our focus was. Once if you define this eye field, then the stem cells have the capacity to self-organize, to form the structures that innately develop. Yeah. And if you look back, retinal organoids, what people do is that they take iPSC cells, forcefully differentiate them to retinal organoids. And if you see the organization in there, which is not like in vivo, because the photoreceptors are facing outward, facing basically the air, and meaning that there is no proper signaling is patterned. So that's what we believe.
It completely mimics the
Well, we tried. I don't think we completely mimicked it, but we tried, and eventually we do see something. It's seemingly mimicking, and of course we can talk about it when we move on to the other slides. Okay, good. So that's about one of the examples of the high next generation organoids, and then the organoid business is a little bit problematic because you have heterogeneity between the one organoid to the other organoid, and then how many can you generate? 20, 30, maybe 50, but that's not going to helpful for our drug screening studies and the large statistically significant data to generate. Therefore, we thought we generate another kind of organoid. We call this as a high Q brain organoids, meaning high quantity and high homogeneity brain organoids. So what we do, we collaborated with the micro patent, micro well farming companies, and then we instructed them to generate inverted pyramid-like structure, like microvels, and then we grow the brain organoids already into that. So basically, we differentiate brain organoids in a confined space. Therefore, you can restrict the size already, and we could do that in the order of hundreds to 1,000. And what you do, you move all of these brain organoids into the spinner flask. And then you culture them over a time, and you can see over a time, like in this case, IMR 90 line, from day 15 to day 30, 50 to 80, like it increases in size. And the size difference between the organoids is significantly less, or there is no size difference. So meaning that you could generate brain organoids in a precise size without a lot of variations, especially if you want to go for disease modeling or drug screening, which is a very important aspect, because otherwise you will have different results with every individual organoids. So this is something that we took it very seriously and we developed it. And that's how our organoid factory looks like. So the spinner flask, every dot that you see is basically, I could see it nicely here, but it's not very visible. What you see, those pearl kind of stuffs, basically they're single individual organoids. So you could generate 1,000 organoids on a range of 1,000 organoids. So if you have like 5 or 6 flasks running, you're speaking about 6,000, 7,000 organoids. From every flask, you can get around 6 grams of brain tissues. Yeah? And you can leave it for a long time. So we have gone up to 180 days and so on. By using this, we can do biochemical experiments. We are now trying to generate patient-specific organoids from Alzheimer's patients and so on. And we can also do some drug screening studies that I'm going to discuss with you. Yes.
In terms of the the complete composition of the brain organoids person adding this, does it also impact the earlier version of the brain organoids? You know what I'm saying?
It's a more similar. I would say it's much more similar, much more relevant to early embryonic development than the later steps. So the organoids that you are speaking here, they do have mature cell types like cortical neurons, myelinated neurons, and they don't necessarily recapitulate the mature brain. Because mature brain has different kinds of cell types that are also coming from non-ectodermal lineages. The early stage brain, I would say, they're mimicking much more like embryonic stage, but at later stage, it is not really mimicking the mature brain, but still you do have relevant cell types that eventually they mature. For example, tau positive neurons, cortical neurons. But that's the current status. Now we want to also improve it further, that I will explain that to you in the future slides. All right, so now I discussed with you about centrosomes and primary cilia, brain organoid as a tool. We think it is recapitulating human brain biology. So we have got all the tools in the hand. We know the molecules we want to target. We know the systems, the model systems. Now we can try to address more mechanistic questions. And that's where I would like to discuss about cilium checkpoint that we lately discovered in the last 5 years. What is cilium checkpoint and how this develops, how this helps, how this regulates the human brain development. Now, I discussed with you in the first slide, this is a mitotic cell. After successful mitosis, the cells exit the cell cycle. Then when the cells are exiting the cell cycle, well, the cilium is assembled in the G0 where the cells are exit the cell cycle. The cilium is a macromolecular structure. 5 micrometer in size, and it's a large, it's a polymer of tubulin, and it's a microtubule-based structure. And when the cells re-enter into cell cycle, the cilium must be disassembled. Otherwise, it is incompatible with the spindle apparatus. You don't want to have a primary cilium in the dividing cells. So you have to disassemble the primary cilium in a timely manner. So, and if the cells are not able to disassemble the primary cilium, the cells get stuck in the G0. And that we call this a cilium checkpoint. For students, if you look at the textbook, cell biology book, there are so many checkpoints have been described. Anav is promoting complex, mitotic checkpoint. In contrast, cilium checkpoint has never been studied in detail. And now if you think about it, the time duration that is required for the G1 to S transition, it is purely depending on the ability of the cells to disassemble the primary cilia. If the cells are not able to disassemble cilia, you get stuck. It's like a traffic light. You get stuck there before the cilium is disassembled. We call this as a cilium checkpoint. Now, how? Yes.
Sorry to bother. I have a question. So if you mutate the cilia during the development of a mouse embryo, what happens?
The mouse embryo will not develop.
Is it because of the neuronal defect
It
or
depends.
embryonic defect?
Well, it depends who you speak to. If you ask me, it's because of primary cilia, I would say. Because primary cilia also, it's a key for regulating the hedgehog signaling, which is fundamentally important for any organism development. Yeah. But for the second question, how it regulates the neurogenesis, I will just come for it. Okay, so how did we discover it? I mean, the cilium checkpoint. And that's what called as scientific serendipity, because until that point, 2016, I had no clue what these genes that we have cloned and we worked in the past is doing in the human brain development. One of our favorite genes is CPAP. And by collaborating with human geneticists, we found a patient who has a mutation in the CPAP. He's a collaborator of us and with Dr. Parveen. He's in Saudi Arabia, Fausan. He's a very famous human geneticist. So he found a patient having a mutation in the CPAP. Okay, so we got the cells from them. And then, these patients present microcephaly as a phenotype. So what is microcephaly? Compared to normal human here, you can see the microcephaly brain is small, but you have got all the structures there. So meaning that the stem cells were depleted during the brain development. That's what we thought in the beginning. Okay, so we teamed up with those guys and we got the cells back to the lab. And until that point, my lab was a Centrosome lab. So by accident, we looked at the primary cilium just because we had some antibody there. We looked at it, compared to the control cells, microcephaly cells had a very long primary cilium. And that changed our life. Don't know if it's good or bad, but that changed. Well, we thought, well, these cells have a problem in primary, disassembling of the primary cilia, or if they have a slow cell cycle progression. So that can be tested in a very simple experiment. What you do, you take the fibroblast, patient fibroblast compared to the control, you serum starve them, you grow them without serum. The cells are 100% of the cells are now ciliated. Now we add the serum, the CPAP protein, the pink color that comes up as a cloud at the base of the primary cilia, and that cells are now disassembled, and these cells re-enter into the cycle in 24 hours. The microcephaly cells don't respond for the serum addition. They're always ciliated, they always get stuck. That's sort of a clumsy and lazy. They get stuck at the long time. Okay, good. Now,
what is the control here? What cell type?
Control is, it's a wild type fibroblast. No, it's not a sibling control, but it is a wild type fibroblast because we don't have the sibling for that patient.
Okay. Is it possible to make that as a wild type?
You mean the microcephaly cells?
No, so you have these mutant cells, right?
Yeah, yeah.
Can you rescue?
You could, yeah, of course, you could rescue it. Yeah, we have done those experiments already in this paper. So then we wanted to do biochemical experiments to understand what's the mechanism behind it. So what we did is that we purified. I'm not going to go to the details, but we purified CPAP, and then we identified a complex of proteins that are interacting with CPAP, and CPAP is providing a scaffold. What it does is that during the cell cycle re-entry, when you add the serum, the cells re-enter into cell cycle. CPAP provides a scaffold. Now these complexes are recruited at the base of the primary cilium. The cells are disassembled, the cilium is disassembled and cells re-enter into cell cycle. When the CPAP is mutated, these complexes are not recruited at the base. As a result, the cells are always ciliated. Now, it turned out to be that, well, this list is not exhaustive. We have much more new components, and mutation in these genes also cause microcephaly via perturbing the cilium disassembly. Okay, so whatever the experiments that we spoke about right now, it's all being conducted in the fibroblast. And how is it all relevant to microcephaly and brain size control? So therefore, we started generating human, for obvious reasons, you have no access to the patient samples, I mean, the brain sample, which is precious. So then we just started generating human brain organoids, which I explained to you a few slides ago. And before we go into the details, and we think this is what our hypothesis, I would like to explain that to you. As I mentioned to you, the stem cells have a unique characteristics of undergoing symmetric division and asymmetric differentiation. For the symmetric division, meaning that the one NPC give rise to identical cell type, which is called as self-renewing NPCs. What we think is that the self-renewing, and for the self-renew process, you need precisely regulated G1S transition. And that is, again, in turn regulated by the temporal cilium disassembly, meaning timely cilium disassembly. If you delay the primary level, and then comes a signal that triggers these cells to generate neurons and then NPCs. So meaning that this population is very important during the brain development. So if you lose this population, if you deplete this population, you don't have enough stem cells. As a result, you don't develop the brain in a structurally normal way. And what we think is that if there is the delay in the cilium disassembly, as we observed from the microcephaly patients, and the cells don't seem to like it, so they prematurely differentiate into neurons. And as a result, you lose this pool. So this is our hypothesis. And if you do have questions, just ask me before we move on to the next slide. Well, first we tested this hypothesis in a 2D experiment, which is fairly straightforward. What you do, you generate the NPCs from the wild type healthy patient samples, and then and culture the cells under the conditions that do not favor the differentiation. That's important. And you can see the control cells, they all have, they all underwent predominantly symmetric proliferation, and there is no neurons. But if you do the same experiment with microcephaly NPCs, you start seeing already neurons, meaning that these cells have a selective pressure that they don't want to continuously proliferate or self-renew, but rather they prefer to differentiate into neurons. As a result, you lose the stem cell pool, then the brain is not developing properly. And that's what we did in 3D experiment. We generated brain organoids from the healthy patient and then the microcephaly patient, and you can see the pink color that is basically NPCs, progenitors here, neural progenitor cells that are drastically depleted in the microcephaly, leading to cortical thinning. So this is what we observe in the microcephaly patients. We also don't think this is the only mechanism is causing microcephaly. This could be one of the major mechanisms. Now we are investigating, basically recruiting massive number of patients in Saudi Arabia who have a different kind of microcephaly. That's the collaboration. We do it with Shaf Kufta. Yes. So in summary, what happens is that the G1S transition is critically maintained by the timely cilium disassembly, and that is when a time clock is operating. So G1S transition, as long as the cilium is disassembled in a timely manner, the stem cells proliferate and expand their symmetric pool, and that allows the formation of the normal sized brain. And if there is a delay, the cells don't like it, they don't self-renew, rather they differentiate, and as a result, you have an NPC depletion that could cause microcephaly. Now, we find that this hypothesis is very cool, and also work with cilium is very nice because it's a dynamic structure, which was not thought to be dynamic because primary cilia are not motile. So now we are asking a number of questions based on the cilia biology, and due to the time constraint, I'm not going to go into the detail. Now we can ask a question: what happens when cilium disassembly is accelerated? So if you accelerate, the cell cycle proliferate fast, right? And I'm going to share some data on that. What happens when NPCs don't assemble cilium at all? So I will show you some 1 or 2 slides in the later. And we think there will be a defect in the brain organoids. The third is cool, really. What happens when NPCs override the cilium checkpoint? There is no communication between the cell cycle proliferation and cilium disassembly. The cells don't listen to. Basically, there is a divorce between these two aspects. And what happens? So due to time constraint, I'm not going to go into the details, but we can talk about it. But the first aspect I can speak about it. I mean, well, so far, cilium checkpoint seems to be a fate determinant and controls the neural stem cell homeostasis and brain size. So we have some data that if you accelerate the brain, cilium disassembly, you expand the pool. So NPC pool and the brain organoid sizes are different. Now we would like to go into this direction. And next question, as I explained to you, that we are interested in translational. I mean, is it possible to target cilium checkpoint in the glioma stem cells? So why are we interested in glioblastoma multiforme? Such a devastating cancer, there is no therapy. The median lifetime is about 1.5 years, and the recurrence is 100%. So by collaborating with the clinicians, so we realized that glioma stem cells have their very similar properties like neural progenitor cells. They all express neural stem cell markers. They self-renew. Only difference is that they don't differentiate, right? So we have a team of members in the laboratory. So what they do, they have a direct access to the patient. The moment they isolate the brain, in the next 2 hours, the sample is in the lab, and we isolate the glioma stem cells, and we look at it. Those cells, at least a major population of glioma stem cells, patients do not have a primary cilia. There is suppressed ciliogenesis. That gives an advantage for the stem cells to proliferate very fast, and they don't differentiate. The idea is that can we introduce primary cilium in those cells, and then eventually first stop them to proliferate, and second to trigger them to differentiate into neurons and astrocyte. Therefore, the tumor volume can be reduced, and this could be a potential target for the glioma medicine. So just to give you a picture that, compared to NPCs, this is from one particular patient that, those cells do not have a primary cilium. I mean, there is only a couple of % of the cells are primary ciliated. Now we wanted to understand what's the mechanism behind it. So as I explained to you, the CDC, cilium disassembly complex, which are forming, but are not recruited in the microcephaly. As a result, cilium is not disassembled. But in glioma cells, these proteins are highly upregulated. So meaning that when the cells are, when the centriole is trying to assemble the primary cilium, these proteins are already upregulated. That brings the cilium down, and that allows this kind of situation. Basically, the cells proliferate, shuttle between the interface and mitosis. There is no cell cycle exit. There is only GSE self-renew. The idea is that if you introduce the primary cilium, could you direct the cells into cell cycle exit irreversibly and allow the cells not to re-enter into cell cycle? Then you can reduce the tumor size. That was our idea. So we targeted our CDC components, but it's a lot of data behind it, but I would like to share only one data. In this case, NIC2, which is one of the component of the CDC complex. And when we expressed the inactive kinase, and you can see compared to naive means that it's directly from the the patient cell line. But when you express the NIC2 KD, now they're all ciliated. You are very happy about it. Now you are looking at the fate of those cells, and you can see this is before cilium induction. The red is basically nestin, one of the neural stem cell markers, and that is completely gone here. So the stemness is gone, but now they gain the program of differentiation. So cilium checkpoint is such a very strong determinant. Yeah. So now, cool. I mean, we could introduce primary cilium and direct the cells to differentiate. Now, what's happening? Can we analyze this model system, everything in the 3D human brain organoids? That is a challenging task. But before that, after cilium inductions, GSEs just behave like normal NPCs. So they can undergo differentiation. We don't know whether they come back to proliferation. It's always possible, but at least we could, trigger them to differentiate, and cell cycle exit them, trigger them to differentiate. Is it possible to model the GSE's invasion in human brain organoids? Yes.
how in this disease model, how did you mimic the tumor microenvironment? Because as far as I understand, that also plays an important role.
That's correct.
Right.
This is much more reductionist approach. I could share some data to you later. We're also now trying to do this not in the cells, but the tumoroid. So we generate the cells, we generate the tissues, we get the tissues from the patient. So then we generate tumoroids, not organoid, but tumoroid. So where we can induce primary cilia, either by genetic ways or by chemicals. And that has got tumor microenvironment, as you asked. You have microglial cells, active microglial cells, and all sorts of things. Now, for the clinicians, it is a problematic because the glioma is highly diffusive. So it doesn't form a sort of a tumor you could easily isolate. But by the time the tumor is there, you have because of this diffusive nature, the complete brain is now diffused with tumor initiating cells. So it's not possible. So first, we don't even know the invasion behavior of a patient. The patient invasion behavior is very different from one patient to the other patient. So we wanted to sort of model this. And how do you do it? These are the glioma stem cells isolated from the patient, and we fused them to the brain organoids, and they started invading. And by applied tissue clearing and imaging, and we can study the invasion behavior. And you can see, if you use the two different cell lines, they behave two different ways. Means there's a lot of heterogeneity. The behavior differs from one patient to the other patient. Now, if you can stratify this invasion behavior, then you could stratify the patients. And that would be the first step for the personalized glioma invasion assay, as well as glioma medicine, in my opinion. So I would like to run only one movie. And you can see the green is brain organoid. The red is basically fused glioma cells. And you can see, kind of a cable-like structure, and that is called as microtube, meaning that one glioma cell is connecting to the other glioma cells through this process. And we can quantify this. And based on that, we can actually say that, okay, how aggressive this invasion behavior. So that's the direction we are going now. Now, I promised you that we also did some drug screening using brain organoids, and that's what everybody speaks about it, and that is highly demanding, but it is not easy, in my opinion, because you need to generate large number of brain organoids, and they should also look similar to each other. So we used this glioma model as a test bed. So what you see here, glioma cells are invading into the brain organoid, and you stuck it to the high content microscope, because now the brain organoid size is very similar to each other. So it's reliable data, you could get it. And then we did a pilot experiment by screening 180 compounds with known functions. By doing this assay, the darker the color you see, more the spots, meaning that the drug did not work. Then we look at for 180 different drugs, the white is good. So we just take those cells that organize the compounds that prevent the invasion of these glioma cells. But these are all patient-derived cells. They're all primary cells, by the way. So like that, we identify a number of compounds, two compounds, which has never been used for glioma, but they do have some clinical values. So we are now generating mouse-based data to validate the organoid-based screen and so on. So this is something the lab is moving into this direction. So the conclusion is cilium can determine neural stem cell fate, determine between the self-renewal and differentiation. And cilium checkpoint is clearly a target for glioma therapy. Not only we are looking for high content screen, we are also looking for target-based approach. So the lab also works on structural biology to identify the CDC component assembly. Now we did identify compounds that prevents these protein-protein interactions, turned out to be FDA-approved compounds. We can readily translate them to the clinic because they were already clinically proved. So for that, we have a large number of collaborations going on.
One question. So when you loaded those glioma cells with cilia, did you check for the epigenetic footprint? The cell loses its footprints or?
Yeah. I mean, we did some bulk RNA sequencing data. We did look into that. We didn't go into that in detail because it's not our expertise, but we do see some changes. It's becoming much more of differentiative than self-renewing. That somebody can look at that. That's complete field of interest. Now, I would like to conclude, my talk with a couple of slides. That's what we think we are going to go in the future. Something called as natural selection, putting a lot of pressure, leading to species-specific gene isoforms. So we ask ourselves why human brain is so sophisticated. So the adaptation of an organism by means selectively reproducing changes in its genotype. I don't believe one organism loses the gene and the one gains the gene. I don't think it is possible, but there is an evolutionary pressure that might allow the gene to modify itself through epigenetics or splicing. We believe one of the aspects is splicing. One of my talented postdoc who joined a couple of months ago, he embarked on this question, and we are trying to identify primate-specific genes. Not primate-specific genes, I would say primate-specific isoforms. So lately, we also inherited this technology in the lab. We can do single-cell RNA sequencing. And so we took advantage of this next generation organoids. And now here you see day 30 to day 60, you can see the structural remodeling in terms of plasticity. And every single dot that you see is a single cell that is sequenced 50,000 times, reads. So we can cluster them into different groups. And one cluster was very exciting, C4, where we found a number of neocortex, human neocortex specific genes. And again, I want to be careful. I don't want to say it's a human specific genes, but can be some isoform that are specific for humans. So by applying a lot of computations, carefully filtering it out, we could identify some of the genes. We call them as CCB because these genes have never been studied. CCB is basically my lab code. And that's one example I wanted to share. It's called a CCB79. So we do have this gene in our lower organisms, meaning that, well, it started from this jawed animals. Fish might have it, but we could not find it. Frogs don't have. Basically, when the animal kingdom moved from water to the land, started getting mutations via selective pressure, and then started getting these isoforms specified. The 74, which is basically our ancient gene. And then Stefan identified there is a mutation that allows the splicing to skip. As a result, you have an extended form of this gene, which is present only in the primate. So now by using this, well, we cloned this gene, and then this has never been studied, and it turned out to be a cilia transition protein, meaning that cilium is really lately evolved structure. I'm speaking about like 30 million years. In the past 30 million years. I will not be surprised if there was an evolution in the last 3 million years, right from the Homo sapiens erectus came. So I try to also capture those materials and try to do the evolutionary neurobiology. We also identified 2 patients with a severe microcephaly. We found only those 2 patients across the world. Now Stefan was thinking, wait a minute, now we are trying to understand how this gene functions. Hopefully the story will be out soon. Now we are trying to put the primate-specific genes into the mouse. Well, we don't want to work with mouse. We don't want to kill some animals, but we want to go for organoids, mouse organoids, monkey organoids. Back then, 5 years ago, we were not able to do these experiments. And see, by changing the ciliary structure, by putting this primate-specific gene, then we can study the human neocortex evolution. That's the direction going on. Then Stefan was interested also. The lab has got different field of avenues. Stefan was interested in looking at, wait a minute, I'm going to look at in the glioma stem cells. Strikingly, he found some splice mutations that allows the CCB53. And that is present only in the glioma stem cells, at least 10 patients. We are going to increase that one to 1,000 patients. And then if we can identify, is it a prognostic or can be a new biomarker, and we also believe this can be pathogenic. So you would like to express it in cells and see what's happening.
I possibly didn't follow the last point. Yes, what was found?
So Stefan was looking at different isoforms in the glioma forms, so glioma cells. So what he observed is that 59 is an ancient form, which is also present in rodents When it comes to the human, this is 79. Yes, as I said, that this is skipping. Only in human glioma cells, there is another form present, 53, which is not present in the healthy cells. We think it is like P53. Yeah? Now we call it as a C53. We have CCB53. So we are trying to do experiment that we express that in the cells, control cells, and see that if they can transform. If that is the case, then we can rewrite the cancer biology a bit more. And that explains us why the sophistication comes from with expensive adversity. So in my opinion, the human brain is so sophisticated. I mean, we can't deny that. With the unique modalities, unique mechanisms, but that comes with specific human-specific diseases. So once we are able to focus on that particular aspect, then we can now address certain diseases which are microcephaly, glioma, and neurodegenerative disorders. If I look back the literatures, microcephaly you cannot model in mouse. Full stop. It's not possible because mouse brain itself, it's a lissencephalic. Yeah, it's a shiny surface. Cilium size is different. The cell diversity is different. But mouse brain helps a lot, but you want to study the microcephaly, can't. And glioma, neurodegenerative diseases, I started thinking these are all human-specific diseases. Because if you want to study those diseases in human and non-human models, you have to always induce them. That's not really physiology. That is actually pathology. So we think identifying these human-specific mechanisms, one way to identify human-specific isoforms, I want to be careful if they're not human-specific genes, but human-specific isoforms, then we can nail it down. What is it talking to, who are its friends, and how this regulates the cell cycle behavior, cell diversity, and so on. Yes?
So I have a few questions. So CCB53, correct me if I'm wrong, that was found in glioblastoma sample.
Yes.
That isoform.
Yeah.
How prevalent is that isoform?
Yeah. Okay. So again, it's first of all, it's a two preliminary data, and we looked at only 10 patients and lines. Yeah. And now we are collaborating with Neurosurgery Institute, and they have always mRNAs isolated. Every patient comes, they have mRNA isolated. So we could simply do a QRT-PCR experiment. Look for these three populations. Do you always see the P53 pops up? Here you go. So that's what we are planning to do. That.
And 79 is excluded.
It's always there.
In humans.
Yeah.
In all the humans.
Yeah.
Right?
Of course, all the humans. Yes.
Of course. Yeah.
Yeah. Well, I believe I do have also, but so it should have. The question is that, well, we still did not explore the mutations in that. Yeah, there may be some activating mutations, we would like to first express it in a wild type background. We don't form the tumor, if we don't transform the cells, we can eliminate the P53, which is a bona fide model that if you eliminate the P53 or inactivate the P53, you might induce, you may favor the transformation, cellular transformation. So we have to look at all those experiments. With that, I would like to thank my collaborators, Roberto Palini for glioma resources, and Michael Zattler and Kamir Hadian. We do drug screening studies, and other colleagues and different funding agencies, including Human Frontiers and Deutsche Krebshilfe and Deutsche Akademische Austauschdienst. And I would like to also thank, I'm gifted by amazing lab members. So most of the brain organoid studies have been initiated by Elke Gabriel. Arul, very talented microscopist. I mean, he could image even the elephant under the microscope. I can tell you that. Amazing person. And Anant, who devised high content brain organoids, the high quantity high Q organoids, and Marco for electron microscopy, and other members of my fly group. Stefan and other members, Nasli Khan and others are not present. It's not updated. It's still my old lab. And so, as I said, I'm really gifted by these guys. And thank you for your attention. I'm happy to take your questions.
Okay, fantastic talk. I think two parts, looking at the later part of the glioblastoma, if I just take liberty of asking one quick question. So if you do a knock-in of CCB53, do you possibly build a hypothesis that it will have the damaged ciliary checkpoint?
That's what we think.
Yeah.
So that's what we think. Stefan called me over the weekend and he told me that the cells have completely CCB53.
Yeah.
And he still did not analyze the primary cilia, and I am going to text him today. I think he should be working now.
That would be a great corroboration of your two
Absolutely.
separate findings.
But again, it's too preliminary, and there are some glitches. You're just following our, those. So let's see.
Yeah, no, excellent.
Yeah.
Thank you. Any quick questions for Jay? Yeah, please.
So I have a question regarding the developmental pathway. So you had shown early on that there's the basal stuff and the cortical neurons, and in between you have the radial cells, which have the intermediate cells. What is the role of this CDC and this primary cilia in the determination of the cell phase?
Yeah, I mean, we believe the fast cycling cells, which are apical progenitors. I think CDC, the proper or accurate assembly of the CDC complex formation and recruitment, it is important for the cilium disassembly that should happen in a timely manner.
So do you think the kinetics of this is different for each intermediate cell?
I see. Okay, that's the question. Yes, that's an amazing question. Yes, I do believe that because every cell type in the human body has a different cilium dynamics. And we also think monkey NPCs has different cilium dynamics than human than mouse. So, as I said, the evolution is not biased. You have got all the components, but the amount of components, how much they are activated, that differ depending the biological function. So, if you look at the mouse cilia, it's ridiculously small. And old world monkeys, like we have our temple monkeys, rhesus monkeys, they have a very small cilia. So we think it's the cilium dynamics, not necessarily through CDC, but several other peripheral components. They can regulate the early development. I'm not speaking about postnatal, but early development. We think so. But also the cancers, you have a different dynamics of cilium. So cilium is traditionally thought to be a signaling structure. Well, I respect it, but it has got some other functions, which is one of the most important function is regulating the neural stem cell proliferation versus the differentiation.
Does the length of the primary cilium increase as maturation in the various layers of the?
It's a cell type dependent. That's what we think. The rapidly proliferating cells, take iPSC cells, you have like 2 micrometer cilium. But if you take the NPCs, it's a little bit longer.
No.
And if you take slow dividing cells, it's really like a gear of the car.
Corticals, it's longer?
No, that is completely a different business. Cortical neurons are post-mitotic neurons, and they don't cycle, and I don't understand why they have cilium. So that is another avenue that we should study. Yeah, sorry.
So you have shown the data for the CFAP where the microcephaly, so they have more towards the differentiation, right? So they form more neurons rather than replicate.
Yeah.
So do we have any idea whether the neurons which are formed after that process, are they have similar nature, the functional wise and the size?
So we haven't tested it. It might be different, but we haven't tested it. Yeah, that's a good question.
It doesn't, and when it reaches a certain size organoid, if you want to work on the neurons. And
I do believe there is some critical, interesting insight. For instance, dopamine energetic receptors and some they're also localized at the primary cilia. People have looked at it. In schizophrenia, you have a long, lengthy primary cilium. But these cells are not proliferating cells, so might be the signal. Now that's where possibly the primary cilium is doing its bona fide job, which is trafficking the signaling. So we haven't analyzed that. That's a good point.
I have a quick question. So the brain organoids that you talked about, do they also have the midbrain and hindbrain parts?
Good, very good question. It's what we generate is whole brain organoids. So we don't force the iPSC cells to differentiate into one direction. So what we do, we differentiate from the iPSCs directly into neural epithelium. So we skip the embryoid body development. So embryoid body, what do you have? You have ectoderm, endoderm, mesoderm. If you pass through that way, then you have contaminated cell types, and then you force the neuroectoderm to develop into certain lineages. We don't do that. So what we think is that we let them, like a Waddington hypothesis, we let them to differentiate, and because of the self-assembling property, they take care of themselves, seems to be. By doing gene expression studies, we could observe number of different regions in our organoids, but morphologically, we don't see them clearly because the proportion is very, very less. So that's the reason we got optic vesicles attached to the forebrain. Yeah.
So so every organoid at day 60 would have optic vesicles.
Well,
Most they are heterogeneous, but
not. I mean, it's two different protocols. So in optic vesicle generation, we use retinoic acid in a timely dependent manner. So otherwise, what people do, they take the iPSCs, they put everything together, push them to one direction. So retinoic acid is a paracrine inhibitor. So that prevents, like decade-long experiment has been done. It prevents the optic vesicle to assemble at the first place. So you don't want optic vesicles to be not to be assembled at the right from the beginning. Therefore, we added retinoic acid. We also optimized the conditions, concentrations, and also the time point. So we add them only when the neuroectoderm expands.
Jay, we'll take couple of questions from the Zoom platform. Well, the first question is, while taking GSCs for the experiment, was the role of microglia taken into consideration, since they play an essential role in controlling the tumor microenvironment?
Absolutely.
And the behavior of myeloid-derived stem cells as well as the astrocytes?
I don't know about this myeloid derived cell types, but I can answer about microglial cells. The brain organoid that we use, they do have microglial cells. Don't ask me why. And I don't have an answer because we never started with mesoderm, and BRCA positive cells are negative. We do have some proportion of microglial cells. We don't understand that. But now we are revising our assay in a different sense. So we don't use glioma cells. We are using tumoroid. So when the sample comes, we generate already, I can show you some data. We generate the tumoroids. That has got microglial cells and all other microenvironment. So now we fuse them to the brain organ. So brain organoid has microglial cells. These guys have microglial cells. So we are going into that direction. So we have to still develop the assay further.
So how are the assembloids different from the tumoroids?
No, assembloid means you have to fuse two different kinds of organoids, not fusing one organoid, one kind of organoid with cells, that is co-culture. So tumoroid, you could call it as it is a naturally available assembloid because you have got very many kinds of cells. Yeah. So if we could call tumoroid, fusing the tumoroid to the organoid, we could call it as an assembloid. We don't want to do that because we want to do our own business.
Okay.
Yeah.
Well, quickly go to the second question. What experiment did you perform to check the light sensitivity of the eye-like structures on the brain organoids?
Good. So it's a primitive experiment. So you dark adapt them in a Nivea cream box, for example, and you dark adapt them with the insert inside. And then we expose them to it's RE, retinoelectrogram. This is straightforward retinoelectrogram. And we measured the negative amplitude of the current measured at different intensity of the light. That's what we did. We could also bleach them, because if you keep looking at the bright light for 10 minutes, the vertebrate eyes, you get bleached and you don't have sensitivity for some time. If you look at even this light, this is about 20,000 millilux, this light. So if you keep looking at it in 5 minutes, you are blind. I mean, you need to dark adapt. And that's exactly what we did in this our organoids. So we exposed them for the light, and then we tested immediately after 1 minute. There are no response. And after dark adoption, it slowly developed the light sensitivity. It's a straightforward
Yeah,
REM
I think
experiment.
it's there in your paper.
Yeah, it is all there.
in the paper.
And
Okay, I'll take the third question. This is the last question. The size of the microcephaly brain is shrunken because of the depletion of NPCs as compared to controls. What about the population of astrocytes or other glial cells? Are there changes in these population as well?
Well, at this point, it's a very early stage organoids. At this point, we don't even expect the astrocytes are playing any role. So one has to understand there are different waves of neurogenesis. The first is that NPCs into neurons, and after reaching sufficient amount of neurons, then comes the astrocyte, and comes the other oligodendrocytes and other cell types. So what we are looking at is a very early stage. So we don't think astrocytes are playing a role there.
And
We
what
need
is
to observe them.
What is the day point? I mean, how
15 days.
These organoids are?
Yeah. 15 days. We optimized to study microcephaly at 15 days. 15 days of culturing in the in the spinner flask.
I think we don't have any more questions. So I think thank you so much, Jay, for taking time.
But there are so many questions here. I don't need to speak about it, right?
There are many more coming.
Role of chemicals
So
in
those 3 questions I think we have addressed.
Okay. Right. Well, people can write to me, right?
Yeah.
But there are a bunch of questions here.
There are
Okay.
more
But can I close it? Is it okay?
Yes, I think so. So thank you so much, everybody, for
My pleasure.
coming and attending the talk, and thank you so much. Really appreciate Jay taking time and explaining the data that we have.
Thanks for having me.
Yeah, thank you. Okay.